Good afternoon, everyone. I'd like to welcome you to today's installment of the CINECA webinar series. Today we have with us Melanie Courtot from the Ontario Institute for Cancer Research, and she'll be talking to us about the integration and harmonizing of diverse large human cohort data. My name is Vera Matzer, and I'm involved in the CINECA project on behalf of EBI, and I'll host today's webinar, and I'll also moderate the questions at the end. So before I introduce today's speaker, I want to make you aware of the fact that this webinar is being recorded, including the question and answer session at the end. The video and the slides will be made available on the CINECA YouTube channel and the CINECA website. We've reserved some time at the end of the session for questions and discussion, and I would like to ask you please to write your questions in the Q&A panel, as you can see on the slide. Feel free to already do that during the talk. You'll also be able to do it at the end. So before we get started, I'm going to briefly introduce the CINECA project to you. CINECA is a 4.5-year project, and CINECA stands for Common Infrastructure for National Cohorts in Europe, Canada, and Africa. And it's received funding from the European Horizon 2020 program and the Canadian Institute of Health Research. CINECA's vision is to accelerate disease research and improve health by facilitating transcontinental human data exchange. Now, there are several challenges that CINECA is trying to address. The first one is the challenge of federated data discovery. This is all around how, as a researcher in a particular domain, do you find cohorts of interest? So this could be by looking for genotype, phenotype, or data use. Second step is the interoperable authentication and authorization. After you've found your data, how do you get permission to access it? Once you have access, you might want to ask questions about the cohorts. And in that case, there will be a need for harmonized cohort level metadata. And that's very much what today's topic is about. And ultimately, the aim of CINECA is to enable federated analysis of research and healthcare applications. We have a set of CINECA demonstrators across cohorts in both the research and clinical setting. Now, of course, underlying all of these challenges, there is a fundamental need for a transnational harmonized ELSI framework to address the ethical, legal, and societal issues. CINECA also has an outreach and training program to aid awareness and to build capacity in these areas. And if you'd like to be kept informed of the progress of the CINECA project, I suggest that you follow us on Twitter or keep an eye on the website. So in that case, I will now introduce today's speaker. So we have with us Melanie Courtot. She's the Director of Genome Informatics and an incoming Principal Investigator at the Ontario Institute for Cancer Research. Her team develops new software, databases, and other necessary components to store, organize, and compute over the large and complex data sets generated by the OICR's cancer research programs. Dr. Courtot is passionate about translational informatics, building intelligent systems to gain new insights and impact human health. She co-leads the data use and cohort representation groups for GA4DH, as well as the cohort harmonization efforts for both CINECA, the IHCC, and the Davos Alzheimer Collaborative. Melanie can be found on Twitter, where she often posts about science, equity and diversity, food, and the silly things she or her children do. At this point, I'd like to hand over to Melanie, please.
Thanks, Vera. Thanks for the introduction, and thanks everybody who's taken the time today to come and listen to this webinar. It's really great to be here and see the enthusiasm for the topic, because obviously it's something that I really care about and I'm feeling very close to. And so today I really want to tell you about, I guess, one of the most practical aspects around bringing all that data we are generating together, and specifically for human cohorts. There's a lot of standards, tools. How can we use those? How can we use those together to build a robust, scalable application for human research and impacting human health down the road? Excuse me. So, as I said, I want to talk to you about human cohorts and how that's used for disease research. But the corollary underpinning our work on human cohorts and data integration is everything around fair data management. So those are the main two topics I will touch on today. So starting with human cohorts for disease research. As Vera mentioned, I've been involved in several projects. One of those is the International 100,000 Cohort Consortium, which, as you can see on this map, is really about a coalition, a global worldwide coalition of countries and cohorts, which are targeting majority over 100,000 participants. So you can see, Mexico has 160,000, the French have over 800. We're working with people in Canada, the US, for example, there's all of us, the Japan. So it's really worldwide, and it's really about a large genomics cohort worldwide. The Global Alliance for Genomics and Health that Vera also mentioned in the introduction is less of a pure cohort program, but is really about responsible genomic data sharing. So it's really about setting up policies like the LC policies that were mentioned, but also technical frameworks to enable those data sharing at scale across all those projects, cohorts, and it's very much a coalition of the willing. Anybody can join the GFRGH and become involved in any of the working groups that are developing the standard and specification. And last but not least, this is a CINECA webinar, and I used to be leading the semantic harmonization work for the CINECA platform. Again, CINECA is a cohort project which targets specifically Europe, Canada, and Africa. And at this stage, I think we have around 13 cohorts and just over 1.4 million participants. And as you can see on this slide, it's again very diverse type of cohorts. So we have some of them are looking at disease. So we have cohorts concerned with mental health, for example, like Colas in Switzerland. But we also have a longitudinal cohort, like the child cohort in Canada, which follows pregnant women and their children after birth as well. So very, as you can see, it's very diverse type of data, type of studies, type of questions behind all those projects and all those cohorts. So why do we care? So some of the questions that we want to be able to address once we bring all that data together is really those questions that would be really hard to answer with only a subset of the data. And so for patients, for example, for rare disease, often, we want to know, is there anybody else with the same phenotype? And do we know more about why this phenotype in those people? What's the prognosis? What happened to people like me? What's a person risk based on their own history? So we know that, for example, for cancer, there are risk factors like obesity and smoking. So based on my medical history, what's my risk for cancer? From the side of the care provider, if we understand what happens on a population level, we can better understand treatment, which drugs will be most effective in which individual. And we know that different drugs have different effects depending on the genomic background of individuals. Should we give radiation therapy to this child? Yes, no. We need to be able to answer those. And how can we better monitor people with Parkinson, for example? Pharma and health tech and researchers are also very interested in those type of questions. So from the health tech, can we develop diagnosis? For example, prenatal test is a big one at the moment. There's also all the trend around wearable. If we can collect all the wearable data, can we start preventing disease before the first symptom appear? That would be amazing. The researcher want to understand better, pathways and which pathways are involved in specific type of disease or obesity, for example. There was a work during the COVID pandemic about, given specific parameters, what is the COVID-19 excess mortality? My plant colleagues are looking into the, crops and conditions for nutritional values, and bringing all that data together could help answer those questions. And then all the drug-related question around drug targeting for pharma companies. So based on this, we've built the first cohort atlas with the IHCC, the International 100,000 Cohort Consortium. This is really what it looks like today. So the central pane shows you the cohorts for which we have collated data and harmonized that data. And I will tell you about how that has happened. You have at the moment external link on the right-hand side, the links to the website, but wouldn't it be nice if that were links to the data sets? And I'll tell you about that at the end as well. And on the left-hand side, a faceted search that allows to filter for the cohorts of interest based on the metadata and the phenotypes and attributes in the cohorts. So as I mentioned very early on, bringing the data together, integrating the data and building the HCC core browser is really cool and interesting. But how did we get there? And how did we get to the point at which we had data that was good enough to enable us to do that? And that's the little detour I want to take, talking a little bit about fair data management for human cohorts. You're probably very familiar with those types of figures and number, and this is based on the survey that was done about, what do scientists spend the most time doing? So what do data scientists do? And you can see that 60% of the tasks are around cleaning and organizing data. And, it's possibly not surprising, the number is quite high, but we've all, I suspect, dealt with data that was very heterogeneous, different format. How can we align it, harmonize it to be able to use it down the road? And the second graph that I thought was really interesting is when people are asked, what's the least enjoyable part of data science? Well, cleaning and organizing the data, yet another 60%, because quite frankly, it's not fun. It's a necessary evil, and having ways to ease that and do that better would really streamline the process. I really like that title, which is about sometimes it's easier to rewrite genetics than update Excel. And there's a couple of citations there. And the bottom line is that when people capture data in Excel, there's underlying rewrite of the data where things are interpreted, like March 1 is interpreted as a date, then it gets mangled. And when you try to bring that with gene names, it just doesn't work anymore. And it's proven too hard to update the tool. So instead, we're going to update the biology and we're going to rename the genes. And I thought that was a very striking example of that interrelation between the data, the tool, and what you can do with the data at the end based on the tool. And the citation was quite interesting because, quite frankly, everyone wants to do the model work. I want to apply cool machine learning, and I don't really want to do the data work. I don't want to spend 60% of my time cleaning the data. And I don't think anybody really wants to do that either. And so I really wanted to give you a couple of examples of how we've managed to work to improve the data quality and to produce those fair data, which are findable and accessible, interoperable and reusable. And we know there's more data coming through. We know that it's not scalable to do it manually. And we know that if we want to integrate the data, and if we want to get data from cohorts in Canada, in Africa, in China, we're going to have to be able to federate those data repository for analysis. This is becoming more and more common. People are building workbench for analysis. We know that people are using more and more AI, machine learning, but we also know that if the data underlying those algorithms is not of suitable quality, the results probably not going to be very good. There was an interesting article that came out where AI interpreted the fonts on scans, for example, because they were not given the appropriate context. And we also know that right now, the current data generation efforts just don't support that big level of reuse at a global worldwide scale. I wanted to give a small example of some of the work my team, when I was at the European Bioinformatics, some example of curation, I guess, and data quality improvement at scale. So this is based on the EBI biosamples database, which is a large biosamples metadata information repository. It has, I think, just over 18 million samples information as of today. Anyone can submit samples metadata. There's a couple of examples here. So you see people, for example, PDX for patient-derived xenograft model. So this is when you take a cancer from patient and you put it in mouse to see what happens. And the important bit is that the biosamples database here in the middle then interlinks to all the other repository at EBI. So you can see how it's important to capture the data accurately, but also link to the other resources down the line. And this will allow linkage between the samples metadata in the middle and the assay or literature data in the other repositories. Excuse me. So the problem with the metadata is that if you don't have good, harmonized, standardized, and consistent metadata, it's going to be very, very hard to answer any of the questions you may have. Those are examples of use cases from the Global Alliance for Genomics and Health, GA4GH. If you're looking at, all biosamples for male individual, for example, well, you better hope that male is captured consistently. And we know that that's not the case. There was a paper from my colleague at ENA showing that just in ENA, which is the bubble there, a couple of years ago, they had 74 different ways to talk about male and female. So, you'd better have good ways to standardize that data and make it accessible for querying. And this is exactly what we've tried to do. So if you look at the data set size, so we have 18 million samples. That means 55,000 unique attributes, things like organism, last update, strain. And in combination with the values for those attributes, that gives us 375 million of those key values data points, things like host Homo sapiens, organism, SARS-CoV-2, and so on. And even though the data is very disparate and heterogeneous, we have a lot of it. We have 375 million of those data points. And what that means we can start processing it in an automated way. And when you look at the plot of the curation, you can see that there's redundancy and inconsistency because it's real life data. People are submitting things different way. You're going to have host human, host Homo sapiens, host patient. So you're going to have very much of a diversity of submission. But when you actually look more in depth, you can see that there's that long tail here where most of the data is actually represented by, let's say, the first 50, 60, 100 attributes. And when you look at the numbers more in detail, you can see that 60% of the attributes here, that red bar here, cover only 5% of the key value pairs. So, if you're looking for bangs or your bugs in terms of curation, the red ones are probably not the first one you should look at working on. The green one, which are a reduced set, which cover 95% of the data space, are probably the one you should prioritize. So that's the type of assessment we can start doing with those. Why is it important? So we've built a COVID-19 data portal. Everybody's in a pandemic. I'm just coming out of COVID myself, so very relevant for me at this stage. And researchers want to be able to find all COVID-related samples, want to be able to understand variation and genomics of the samples. And we know that different people will submit things in a different way, again, different naming. So things COVID-19, COVID-19, coronavirus infected, SARS-CoV-2, severe acute respiratory syndrome. And you can try to do metadata curation, semantic annotation, but you also have common challenges in the sample metadata. So things like the character, dash versus no dash, acronyms, typo, and synonyms. And the way we can address those is using automated curation. So we can do things like, dash versus no dash. We can do that automatically at submission time. We can also do manual curation by expert, and we can store those manual curation rules. And I will tell you about that. And then we can build automated curation tools based both on the manual input, so semi-automated tool, and machine learning. And when you start layering all those from your raw metadata, including text curation, semantic annotation, and really the gold standard, the manual curation by expert, you can see how you can really enrich the data and improve its quality. And some of the resources that are well used and well known to do those are the gene ontology or the monarch disease ontology to represent disease, for example. And that's really the example I wanted to talk here about. So this is one of the tools, the Ontology Lookup Service, which is a repository of biomedical ontologies, where ontologies are vocabularies that allow us to represent things consistently. Here's COVID-19. So you can see this is the Mondo resource. And you can click on that link and you can reach that page, and that page will open the Ontology Lookup Service. So you can see that each term has a stable ID, a stable address. Here it's Mondo 01000096. This means COVID-19. We know that it's a type of coronavirus infectious disease. We know that it's a disease, it's not a virus, and so on and so on. So that's the type of information that we can start adding semantically to the data we have. We also know that we can use curation rules. So for example, when the data comes in, we use another tool, which is called Zuma, which allows us to do automated mapping based on what we already know from existing curation. So we know that, for example, SARS-CoV-2, there's a bunch of different related values, which really all mean the same. And so we can train a system to say, well, if somebody submits information with those values, human coronavirus 2019, Wuhan coronavirus 2019 nCoV, we know that what they mean is that term, then they mean SARS-CoV-2, which now has a taxonomy ID, which we can actually assign to the sample. And this is the type of information we can use to consistently retrieve all of the samples instead of saying, give me everything that's SARS-CoV-2, Wuhan coronavirus, COVID-19, COVID-no-19. We can say, give me everything that has NCBI taxon 2697049. And that's really, really powerful. If you look at the COVID data portal right now, there's over 5 million samples which are pulled in from biosamples, which are also linked to other data, proteomics data, literature data, and so on and so on. And that's made possible because there was that work done on cleaning up and standardizing the data. And 5 million, that was not done manually, that was done semi-automatically with curation rules. The last thing that I want to mention about curation, and I hope this is going to work, is when we looked a little bit deeper in the attributes in biosample, we can start plotting them. And this is a video that Matt had done. And we see that you can lay out each of the attributes. Each dot here is one of those attributes in biosamples. And you can look at how do they cluster together and how do they align with each other. And you can weigh them. So the size of the bubble relates the weight, and the distance between the bubbles relay how often they co-occur or not. And if you overlay, if you overlay the labels of some of those attributes, you see here in the middle, latitude and longitude, and surprisingly, they're often together in sample. Things like sex are often represented, but then you can start seeing very interesting things. We have two big bubbles here, an orange and teal one. And when you look at those samples, both of those are talking about human metagenome. I think they're different organisms. I think one is oral and I think one is vaginal. I'm not sure, don't remember. But they're human metagenome data. They're very similar, very close to each other, but they're still different. And that raised the question, is there something we could do here with the human metagenomics community and maybe bring those bubbles together and have a shared standard for shared data representation? So those are interesting things that you can start looking at when you have that wealth of data. We've developed different tools as well. So this is really just as a reference. We've developed a new validator for semantic-based validation, so which will use the ontology in the backend to validate the data. We've annotated our data with bioschema, which is a subset of schema.org. So that means data sets are showing up on Google. And we export all our phenotypic data using phenopackets. Both the validator and phenopackets paper have been accepted. So they will come out very soon if you want more information. Going back, so we've talked about data quality and how important it is to clean it up and having it being amenable for further down the stream cohort-based disease research. So going back to our human cohorts and how did we build the IHCC browser and why did we build it? So the use case starting with was, okay, there's lots of cohorts and they're all collecting data, but as a researcher, I just want to be able to find that data. So I want to find, for example, cohorts that have blood measurement data. Here's an example of two cohorts, COGES, which is in Korea, and GCS, which is in Golestan cohort study, which is in Pakistan. And some of them are using PDF, some of them are using Excel for their cohort information. And the way, for example, they represent glucose and blood measurements are different. So COGES is talking about glucose fasting and GCS is talking about BP, left one, DBP. And for a human or a computer without any more context, it's just really hard to understand what that means and how those should be brought or not together. And so it was really about bringing the cohort data together. It was about having the right data model that would represent both what was feasible with the cohort data, the data access condition, and the cohort data itself, having tools and processes to do the implementation, to do that data cleanup, integration, harmonization, and presentation down the road, and also bringing in those clinical cohorts and making sure that it was all working together. And so the first aspect of the process was around data and models. And I want to tell you today about the data use ontology and the Gecko ontology that I developed. So the data use ontology is an effort under the Global Alliance for Genomics and Health framework. And it's really about describing what's permissible for people that access the data. This is an example from the EGA, the European Genome Archive website. So people can go to the EGA website and they can find data sets they're interested in. And those data sets will have, you can see the little bubbles, the colored bubbles in the middle or in the table are more details. So we'll tell you, okay, this is a data set that's restricted for certain disease studies, only genetic studies, and so on. And so you can understand by standardizing what's feasible or not with each data set, you can understand what you as a researcher may have access to or not. For example, some data sets are restricted for non-commercial use only, so a pharma company wouldn't be able to access those. And so that's what the data use ontology aim at representing, data access conditions for data sets. The other ontologies that we've worked, we started building in CINECA a model based on those 13 cohorts, which we showed you earlier. We built another lap of the attributes that was cohort shared. So for example, a lot of cohorts are capturing demographic information, so the age, the sex. A lot of cohorts are talking about smoking status, did people smoke, and so on, and so on. So we've collated those attributes and we looked at the overlap and we formalized that in an ontology, which is expressed in the web ontology language, OW. And that means it's standards, it's a World Wide Web Consortium standard. We've adopted best practices in the community. It's really very interoperable with other resources. It's available publicly. It's all licensed under CC BY. So really an effort towards interoperability. And I'll tell you a little bit more about that too. And that's what Gecko looks like. And it represents commonly used attributes for cohort metadata. So things like medication, which type of drugs or treatment patient took, which type of genomic data was collected, there's demographic information, there can be cognitive measurements, disease, and so on and so on. This is, and again, the Gecko is available online if you wanted to have a look and even contribute. So now that we talked a little bit about data and models, I wanted to switch to the next part of the bringing it together, which is the tool and processes aspect. I'm talking a little bit about the harmonization process that we've deployed. So, if we look at the pipeline overall, we have at the top left here, we have to collect the cohort data. So we are not collecting the individual level data, we're collecting the metadata. So things like glucose fastings, tobacco use history, age, sex, disease, and so on. So those are the dictionaries we've collected, and we have a little bit of admin data, number of enrollee, PI name, and so on. We export that into all files, again, just to have a standard share format for interoperability. And we plug in a metadata model. So from CINECA and Maelstrom and other international organization, we derive the Gecko model. We bring the model and the files together. There's a mapping step, it's, tooling, process, and pipeline has to run. And then we end up with compliant harmonized metadata where all our CINECA cohorts or all our cohorts use the same attributes consistently across. And then we can export that and make that available for presentation. So a very simple example of the harmonization work. So here, for example, this is a tree from the Gecko ontology. You can see in blue on the left-hand side, we have classes coming from the clinical measurement ontology. The goal is not to reinvent what other people have already done, but rather to extend and reuse, again, for maximum interoperability. So for example, the clinical measurement ontology tells us about blood measurement value and blood glucose level value. And we can expand on those for the specific needs of Gecko. So for example, we know COGES talks about glucose fasting. Well, we can add that blood fasting glucose level value in between, and then we can start playing that jigsaw type of thing where we have an existing tree and we can intersperse the different classes from the cohort and build kind of a mega tree of all the attributes. And that hierarchy allows us to browse the entities much more easily. And an example is here, remember the blood measurement value and our initial use case where the researcher wanted to be able to search for blood measurement values. So you can cut at a specific level in the tree and run your query against that merge view of the cohorts. So when we start thinking about the last part of the process, applying these techniques to the clinical cohorts. So here again, a researcher looking for blood measurement data, but now we know that the glucose fasting is actually a subtype of a blood glucose level value, which is a subtype of blood measurement value, and so on and so on. And you can do the same jigsawing and plug and play for all the cohorts. So for GCS, we know that BP left 1 dBp, that very cryptic term, is actually a type of diastolic blood pressure value, which is a subtype of blood pressure measurement value, which is a subtype of blood measurement value. And you can see how that allows us to align those terms at a higher level. There's been, we started doing the process manually, but we've still refined it to automate. So we've developed pipeline to ingest data dictionary as CSV. And we've developed a pipeline to automate the mapping step, which is the major, that interspeeding and building that mega tree manually is very time and expert consuming. So we've done that. We have a system that allows us to slurp in Google spreadsheets, map it automatically in the middle. And you can see we're reusing those annotation tools like Zuma I mentioned earlier. We get feedback from the cohort, so there's a feedback loop happening here. And once everybody is happy and the mapping is done properly, we push everything into a cohort registry at the end. And that allows us to have all the cohorts in one place with harmonized, standardized metadata attributes. The cohort registry is available online. It gives us ways to access each of the entities. So you have each term is getting a unique ID that is dereferenceable. So you can use that ID to access the registry. There's extra things like change, history, versioning, updating that using standards and tooling standards allows us to do more easily. We've also built a mapping tool. So, we can store the mapping, we can learn from the mapping. If somebody said tobacco use history is mapped to smoking history, we can store that, and the next time somebody shows smoking history, we know, oh, that's actually mapping to gecko tobacco use history. So we can learn from mappings when they have not been done automatically yet. If somebody does manual mapping, we can learn, store them, and reuse them for the next cohorts. And I think that's going to be a big opportunity, as some cohorts already use shared models as well. And together, that has allowed us to build the IHCC Atlas, which is available online publicly, and you can have a look. We're in the process of adding more cohorts. We currently have 12, and I think we're expecting an update over the next month. So final thoughts about, summary, bringing it together, bringing tools, processes, and applications together. So I gave you a very short introduction to the EBI semantic toolkits. There's a video that's linked here that has much more information. Those are all public tool, open source. Anybody can use and deploy local instances. So tools to search, visualize ontology, mapping, annotate data. And again, more information in the video that's linked here. The Overture suite, which is something my team at OICR is building, it's been really tremendously helpful. It's a suite of software modules, if you want, that enable us to do the authentication authorization through Ego. We have systems that do the file transfer and storage, score. Songs allow us to track the metadata that's been collected and validate it. Then Maestro does the indexing, build that into an Elasticsearch index, and that gets displayed in that arranger view that you've seen in the IHCC browser, where you have a search API, the data portal, the faceted search, and so on. So Overture is a system that supports the whole life cycle of building the platform. And we're also building in interoperability with other standards such as GF or GH. We're in the process of providing a self-installer. So instead of having to install the Overture from sources, you'll be able to download a package at double click and we'll just install everything. And corollary to that, we want to add more front-end customization so that people can add their own logo, choose their own CSS and colors and so on. So this is very much ongoing as well. Because we've used shared tools like Overture, we've been able to very easily repurpose the work we've done. This is really just a screenshot from the Davos Alzheimer Collaborative Atlas, which is a different set of cohorts that are focused on Alzheimer, because this is what the specific project is about. But we were able to just reuse a pipeline, customize it a little bit. So for example, for Alzheimer, they were interested in capturing a little bit better the participants' age and the age ranges. So we were able to update the data model, feed that into Overture, reindex everything, ingest new cohorts, and deliver a new product very quickly. And there's a video demo of the process as well. Similarly, the COVID data portal in Canada, the virus-seq data portal, because we've used, again, shared known tools, took us a month to set it up. So from the point in time where we said we need a COVID data portal to delivery of an online portal, it was a month. And we currently have over 250,000 viral genomes as of March. And we're still expanding the project, just extended for new additions. Finally, community engagement is key. I told you about CINECA, GA4GH, Davos Alzheimer. We've worked with many projects, Maelstrom for data model, Elixir, FAIR+. None of this happens in isolation. It's all about working with other people, trying to figure out where the gaps are. They find out gaps in what we're doing. We work together and we improve it all together. The data use ontology is a GA4GH product. It's been a success worldwide, over 200,000 data sets annotated today. CINECA is building a pipeline that relies not only on Maelstrom and Gecko, but also on Beacon or the GA4GH Discovery Search API, the data use ontology, the passport standards. So those are all GA4, GH standards. And again, that means we're interoperable at scale. And very finally, some of the work we're doing in GA4, GH for cohort representation. We've surveyed people, they've reflected on some of the pain points. Lack of interoperability, for example, was a big one. And some of the requirements where people wanted a computational description of the cohort. And that's an active group that you're all invited to join if you're interested. The goal is to be able to find cohorts. So I want cohorts that capture blood measurement information, but also being able to search within cohorts. So I'm looking for female asthmatic patient with a certain level of creatinine in blood. And so those are the two use cases we're tackling here. The very first one, the cohort registry discovery about finding blood measurement data. This is very much the use case the IHCC browser is fulfilling, where you can filter for blood measurement and it's going to tell you which cohort are capturing that information. The second use case around looking inside one cohort is what my colleagues at HDR UK are looking at. And you can see that the diagnoses, for example, are inconsistently represented, for example, between SNOMED CT or ICD-10 nomenclature. And they're looking into that type of harmonization to be able to search cohort by lab results, for example, and ranges of results. And we're working with GA4GH to bring those two aspects together and have something that can be done across cohorts, but also look at values and ranges to search for. And finally, what we're going to do, bringing the data together is obviously critical and really important, but it's not the end of the road. The end of the road is providing that for researcher, providing the data from the catalogs here in the middle, providing the data to users in workspaces so that they can run analysis and get the results here at the end. And that's a project that Nikki Mulder is leading on that was just funded in September 21 to build a data science platform in Africa. That's all I wanted to talk to you about today. I'm happy to take any questions. And I think, thanks to my funder at OICR, and I think there's a slide I'm handing back to Vera now.
Thank you very much, Melanie, for a really interesting presentation. If I can ask everyone to add their questions to the Q&A panel. There was also a question about whether the presentation will be shared, and the answer is yes. We will put both the webinar online. It'll probably take us a couple of days just to get it into YouTube, and then you'll be able to find it both through YouTube and the CINECA website. So we already have our first question from the audience, so let's dive right in.
Stop sharing so I can get access to the Q&A panel.
Exactly.
Everything disappears otherwise. All right.
So the first question is, thank you for a very informative presentation. You've talked about harmonizing the metadata, but could you also touch on how you conduct the data harmonization?
Thank you. That's an excellent question. So the simple answer is that at this stage, we have not done any data harmonization. That's actually something that our collaborators at Maelstrom have done, and they've done really successfully. I think the easiest way to look at it is that it's two ends of the spectrum, where on the metadata side, we've been able to process a lot of the metadata using automated tools, curation, bringing that together. The data harmonization requires much more expertise and kind of hand-holding. So the Maelstrom team has a dedicated curation force that allows them to do that data harmonization for specific use cases as well. So on the other hand of the spectrum, they're very specific use case driven, and that allows them to dive much more in depth on the data point. I think so. The data harmonization is definitely something we want to do. I think the way we will be able to do it is building a middle layer from the high-level metadata harmonization to the specific data, maelstrom power data harmonization, having some sort of middle layer to do the data harmonization, but possibly not quite as granular as they're doing.
Okay, thank you very much. Then maybe another question. You've presented a number of tools that will help in this metadata harmonization process, but what remains to be done on the tooling side?
Thanks, Yura. That's another really good question. I think a lot of it remains to be done. So I think if I had to say, you know, short term, medium term, long term, the short term is on the Overture platform. We are looking at a data access tool, adding a data access tool, which would allow to match those dual codes, that describe data use between what a researcher wants to do and what's allowed on the data set, and does that automatically so that you could try and start to streamline data access. And similarly, adding other standard like the GA4GH passport for authentication and authorization. Then medium term, I think there's going to be a lot of work on, first the data harmonization, but also interoperability with other projects. So for example, I talked about HDR UK. So we'd like to have, at least at the portal level, have interoperability so we can jump from one to another. And I think other people like BBMRI, Eric were interested in that, Maurice Svert's group. And longer term, I think it's really about the analysis and building those secure workbench that people can either download the data to, summary data, or have the way to do federated queries locally. So for example, the Japan data, the genomic data, they're not allowed to take it out the country. So it's really about bringing the analysis to them instead.
Okay, thank you. Then maybe if we think from the point of view of both the researcher, but also the institute and funders, what would you say is the one thing that people could do to make data harmonization efforts easier?
So the very practical, I think very easy possibly step is provide data in a computational amenable way. We've had data in PDF, which really is not fun to work with and extract and load and transform. But I would say, if you generate data or if you have to store data, know your data, capture, everything you know about your data. How was it generated? When was it done? Who did that? Did you apply any transformation? Did you run any software on it? Which version? Everything that you know about your data that you could, give a data bucket to somebody and they'd be able to reuse it. Everything you can think about, I think would be really, really helpful. There's that saying, which is metadata is a love note to yourself in the future. I think it's really that we've all changed position, changed lab, people have come in our team, left our team. And when you start looking at data that's 5 years old, you're like, oh my God, what did I do? How did I get there? Even yourself is really hard to remember when whether it's data or code is all the same, right? So, yeah.
That sounds very familiar, so I think that's very good advice. So if I can ask the audience, if you've got any remaining questions, please add them to the channel. Maybe something that we can, from the CINECA side, already tell you about is that we are currently developing a training workshop on data harmonization, which will come out in the next couple of weeks. And the plan is also for that to be kind of integrated in a learning pathway going forwards. So besides a more general CINECA learning pathway on federated data analysis, there'll likely be a bespoke one on data harmonization efforts as well. So that's something to keep an eye out if you're interested in this subject. And then I think because we are getting towards the end of the hour, it remains for me to say thank you, Melanie. That was incredibly interesting. So thank you for being here today. And thank you to the audience for coming. And we hope to see you back in future CINECA webinars.
Thank you.